Shall we start? So, hello everybody, thank you for being here. Today it's a pleasure to introduce you to Stéphane Avril. Stéphane is a professor at Mines Saint-Étienne, a Grand École d'Ingénieurs in France. He's the director of the Symbiose unit, which is a mixed research unit between École des Mines de Saint-Étienne and INSERM, which is the National Institute of Health in France. Stéphane is originally from Saint-Étienne. He studied, he's graduated at École des Mines de Saint-Étienne, did his PhD there. Then he moved to Arts Métiers in Paris for some time, working as assistant professor. And then he moved back to Saint-Étienne, where he started working a lot in biomechanics, modeling, stent deployment, blood flow, influence of compression stockings, mixing, computational mechanics always, an experimental method, even developing their own testing methods and their own devices. And lately he has moved more towards mechanobiology, towards the smaller scales, cell mechanics, etc. He has led big projects like an ERC consolidator run and more recently ERC proof of concept. He has created 2 startups, many publications. He has supervised many PhD students. From my personal side, I know him since 2018. I joined as a postdoc in his lab. And then I stayed there for some more time as an assistant professor. So he was a colleague there. And he's been a great mentor for me. So today he's going to present his research, and also he's here because we are starting for 1 year. We'll have, well, for the next 3 years, we'll have a joint project between Lacan and Mines Saint-Étienne to start a collaborative structure between the 2 groups. For that, we have Beatrice Virgini, who will be working for a year here between the 2 of us. So thank you very much, Stéphane, for being here. And
thank you, Michael. It's an honor for me to be here. And yeah, as you said, I come. Laser is not working. Okay. So I come from Saint-Étienne, close to Lyon here. And this is in this Auvergne-Rhône-Alpes area. And this area, they gave us a funding to have this collaboration with Michael. They have a council and they also support research. And it was, I mean, it's very competitive because everyone wants to have this funding because all the funding, they look easy to get, but after it's everyone wants to have. And we tick all the boxes actually because Catalonia is a twin region for Auvergne-Rhône-Alpes. So they, and also our school, Mines Saint-Étienne, joined the Time Alliance, and UPC is also in Time Alliance thanks to Diego, by the way. And so it was another thing, and so there were many things that supported our application. So we got the funding, and so we have Beatrice working on this joint project, and yeah, this made me be able to keep a link with Michael. I was really happy to work with. So today in my presentation, yeah, I'm going to take you through a journey about what we did related to thoracic aortic aneurysms in my group. And so I just have, this works. Yes, okay. So this is just a picture of this Symbiose research unit. So this is a joint unit between different institutions. So the institution I work for is École des Mines de Saint-Étienne. So this is a school of engineering in France. But we have this research unit where we have also INSERM supporting us, and also the General University of Saint-Étienne, which has the Faculty of Medicine. So our building here is where we are only engineers. And then there's a hospital. So it's very interesting for us because the only engineers in all this area are located here. And all the other engineers of our School of Engineering are based in another campus, which is 10 kilometers away. And actually, this is another picture of our building. And we have 4 groups. One in soft tissue biomechanics, and the 3 others are in biomaterials, inhaled particles, and engineering of care systems. And engineering of care systems is more related to modeling the flows of patients in hospitals. So my group is the soft tissue biomechanics, and we have different topics of research, but the one I'm more involved into is related to aneurysms. And here I'm going to talk more about ascending thoracic aneurysms, but an aneurysm is a disease, and I'm also going to talk about also other parts of the aorta. And this disease, it's dilatation of the aorta. So the aorta should be like a pipe with like constant diameter, and when it dilates, it's not normal, and it's related to some changes in the composition of the wall of the aorta that is irreversible, and also that leads to some weakening. So the weakening means that the strength is going to decrease, okay? But at the same time, the load increases because you all know the Laplace law. So if you keep the same pressure, but you increase the diameter of the tube, then the strength is going to increase. You increase the stress and you decrease the strength. So there is a higher and higher risk of rupture. And rupture means that the blood would go into the thorax, and then this would be 99% of people would die, except if this happens like already at the hospital. Okay, so this is very serious complication of this disease, and also very biomechanics. Okay, so I mean, I'm talking about stresses, strength, forces. So that's why we were really interested, and a lot of people before us were always interested in understanding more and more these diseases. And you will see that actually, the more you try to do mechanics on that topic, and the more actually you see that also mechanics has its limitations, and you need also to include biology. So the first goal we had, and I think more or less was around 2010, it was to develop computer models to assist surgical interventions on the aneurysms. So just you have to consider that usually a large number of people who have these aneurysms, they will be detected because it's usually related to other complications. So some people, they feel the heart has some issues, they go to the cardiologist. The cardiologist would propose an exam, and they detect the aneurysm. Okay? And they detect it, and then with different imaging techniques, they would be able to measure the size, okay, by the diameter. And if the size goes beyond a given diameter, which is 5.5 centimeter, then they would propose the surgery, because they know that the surgery has also some risks, okay, because it's a heavy surgery, but they know that it's a higher risk statistically if you keep the aneurysm. And so one of these aneurysms is for the abdominal aorta. And I was working with a surgeon, and he said, okay, we have a new type of surgery for this aneurysm. It's a non-invasive. It's we bring the stent covered with a graft. They call that a stent graft, and we can deploy it, and then this will just avoid the aneurysm to be pressurized. So could we model that? And so he said, and could we do something like we see, okay, there are a number of people who are creating startups in the world, and they simulate surgery. So that in 2014, there was like Charlie Taylor from Stanford University. He founded HeartFlow, and this company, they got directly the FDA approval, and they were on the stock exchange and so on. But there were other companies like FIOPS in the Netherlands, and so we said, okay, can we go into that direction? And I said yes, but well, in the end, it was only half of me really involved in that adventure, because I mean, the needs of a company are a lot related to business. But I'm going to tell you actually what it consists. So the company name was and is still pretty certain. Now they are quite successful. They employ 30 people, and actually they do a finite simulation of this device. So this is the stand covered with a textile that is made of PTFE usually, and simulate the deployment of that inside an elastic aorta. and then predict the location of these fenestrations that you see here that needs to be located precisely at the same location as the branches that you have. So this is like a renal artery here, and this is a mesenteric artery, and these arteries, if you prevent the blood going to this artery, then you would lose some your kidney, or you would, so it's very important, and every people is different, and it's very difficult to make this whole fit exactly at the correct location. So finite element modeling was very useful, and now they provide this service to hospitals and to stent manufacturers. But I can tell you, and I know I heard also different other people who went on these adventures of creating companies, the technology that they use is a technology that was already available 20 years ago from now, okay? In the finite element software, it was already used in the automotive industry, and there's nothing really that came out of our research in biomechanics, okay? And I thought, okay, what are the challenges actually related to biomechanics of these aneurysms? And the challenges were more on the scientific side to be able to use computer models of their growth. Okay, so how could we predict if they are going to grow or if they are going to be stable and so on? So this was more like the challenges that we wanted to work on. And so thanks to the surgeon and the collaboration with the hospital, we had access to a lot of images, and some of these images are based on MRI, others on CT scans. But one of the MRI, with the MRI, we can measure blood flow. This is called 4D flow MRI. And with the CT scan, we were able also on some patients to have 10 CT scans and 10 different times across the cardiac cycle, so that we could measure the motion, precisely the motion of the aorta. And from the motion, we could track the displacement of every point in the aorta, and then for every point, then the strain, and then locally we could also get access to the stress by deriving the Laplace law and the pressure, and then just like deriving a stiffness parameter. And so this is here for a patient with stiffness distribution that we found, and you see that you have a hotspot of stiffness here. Okay, so the stiffness parameter is just related to the local stress-strain relationship. And for the same patient, we had a 4D flow MRI. So with a 4D flow MRI, during the cardiac cycle, we had 20 estimations of the velocity vectors inside the artery. But it's very difficult to use them because the spatial resolution is not good enough to estimate the wall shear stress close to the wall. Okay, so what we did is that we used the velocity vector at the inlet as boundary condition, and we ran a CFD analysis in the aorta with these boundary conditions. And then we estimated the velocities, and we worked with colleagues from Politecnico Torino, and they proposed us to derive a parameter that relates to the residence time of the velocity vectors close to the wall. And the idea is that actually locally you have a wall shear stress, and it is a vector. It has two components, let's say one in this direction and one in this other direction. And then the velocity vectors, actually, they are going to have different directions, and in some locations they may vanish. Okay, so this is like a fixed point. And then we looked at where we had the 6 points, and what we noticed is that they were located at the same region that we have this stiffness here. And so there was a combination of factors that actually these 6 points, they correlate with the stiffness. So we thought, what is the link between these two things? And the thing is that these 6 points, they will correspond to a lack of mechanical loading on the cells that are making the interface between the blood and the wall, which are called the endothelial cells. So these endothelial cells, usually, they will have the blood applying a wall shear stress, but in some regions where the flow is disturbed, then this will lack. And then due to this lack of mechanical loading, then these cells, they will emit some signal, and the signal that relates to an inflammatory response. And this inflammatory response will cause different over-signaling that go inside the wall and create a change of the regulation of the maintenance of the structure of the wall, and especially with the production of enzymes that degrade one of the major proteins that we have in the wall, that is called the elastin. So, so then this triggers a process that I'm going now to describe, but we discovered that by just comparing these two things. So what is the process that is triggered? Actually, another observation that was done actually by a group in the US. They found that they used also this 4D flow MRI, and they found that when you have an elevated wall shear stress, then locally the wall has a fragmented elastin. So in the wall, I didn't describe, but you have different layers, and in these layers you have different proteins. And one of these proteins is elastin, and it behaves like rubber. Okay, it's the same behavior as rubber, and if it degrades, then you lose the elasticity. Okay, so they found that this protein was fragmented and not fragmented in the regions where there was a normal wall shear. So we were a bit puzzled by these results, but we understood that actually these fixed points are always located at regions where at the fixed point you have no wall shear stress, but all around you have very large wall shear stress. This is just like because there is a high gradient of wall shear stress in this region. So it's exactly the same observation. Actually, the blood usually, and these aneurysms, they occur in patients who have usually an issue at their valve, this valve that opens and closes at the from the heart, and then this creates a jet, and the jet impinges on the wall, and this is the region where the elastin damages. And so this relates also to this inflammatory response that is triggered by the wall shear stress. And we also found the same also with another PhD student in the group. And so we modeled it like this, okay? So when we have this jet and this wall shear stress that is disturbed, then the initial stress strain response that the wall has, that would look like this, and this is the set point where the wall would be for a normal pressure, an average, okay? Then due to this inflammatory response that is triggered at the endothelial cell level, but then that induces the damage inside the wall, then we have a softening, okay, this is what damage does, but also an increase, okay, of the stress, because the softening of the same pressure will dilate the wall. And then the cells inside the wall, so another type of cells that are the smooth muscle cells that are inside the wall, they will have this overstretch. And so they will, again, signal that, and then they will create a new protein, that is called collagen. So it already exists in the wall, but they will renew it and create more so that this can compensate for the damage of elastin, and this can reduce the stress that these cells they feel, and then make the stress become lower. But by replacing collagen by elastin, then we do not get back to the initial state, and we have a residual dilatation. This is why we have an aneurysm. Okay, so that's the main process that occurs in the wall, according to our model. Actually, that's and so we coded that in as an abacus UMAT, and then we applied that to the geometries of aneurysms that we got from the patients, and actually these results I show it well only the result, but I could do my own seminar, and it's a shame because I think that's maybe the part you would be the more interested in, because I mean in a computer center, but there were a large number of issues because the model that we use here is a hyperelastic model. That is a mixture. So we combine in the hyperelastic potential. We combine different potential modeling collagen and elastin, and each collagen family that we have inside has a different pre-stretch. And collagen is usually modeled as a 1D material that is embedded in a 3D matrix. And this 1D material has an exponential behavior. And then the 3D matrix around is elastin, which is modeled with a Neohukian material. And we encountered a different number of issues also related to the incompressibility of the medium that we have to assign, and to the different, let's say, heterogeneities that are created by these changes that we simulate with time. That a lot of time we ended up with some divergence issues in our model that took very long to obtain such results like this. Okay. And I'm happy to discuss with some of you about this, because this is still a number of things that we're still working on that. And well, the help of specialists like you would be great. So, but the thing is that but when we did that, actually, we didn't really take into account what is really going on in terms of mechanobiology, because mechanobiology is not like observing modeling the phenomenologically what's going on, but it's also maybe understanding if the cells that we assume they would have this response, actually, if they do really have this response. And so we wanted to understand more why the cells would, and also why an aneurysm is so different for a person to another. How can we be patient specific? Because what I did here, what I showed here with this model and this softening and then the replacement of elastin by collagen is something that we don't know how to separate someone who will have a genetic disease and will not be able to produce more collagen or less collagen. So we wanted to have more insight into the cells. So this is more recent, around 2018. And I give you here a better picture of how the wall of the artery is it works. So actually, it's a laminated structure. So you have, for instance, in the aorta, you have about 70 of these different lamellas like this. So the gray part here is the elastin. So this is a thin rubber-like plate, and between you have the cells, these smooth muscle cells. So the one that will start to produce collagen when they feel that the stress is getting higher. And the thing is that these cells, it has been observed that in some patients where the aneurysms they occur, most of them, they have a problem to these cells. They have a genetic disease. People who have this genetic disease or animal models where there is a knockout to some genes, then they automatically produce this aneurysm. One of the examples is that, for instance, in these cells, you have a cytoskeleton with you have fibers that relate where the cell is attached to its matrix and the nucleus. And these fibers are always stretched, and they are made of two main proteins, myosin and actin. And it has been observed that if there is a genetic disease related to myosin or to actin, then you can imagine that these fibers will not work. But all the time, aneurysm. All the people who have this genetic disease, they develop an aortic aneurysm, and all the animals with this knockout, they develop an aneurysm. The same for the attachment to the matrix. If there is a defect of one of these protein aneurysm. So it has been observed that what is called this mechanotransduction axis that enables like the transmission of forces from the matrix to the nucleus, if it is broken somewhere, then an aneurysm would develop. Okay, so how could this be understood and taken into account in our model? Because now, for instance, well, we do not take that into account in what I showed. So we started to consider, okay, if someone has an aneurysm, we don't, and there's no genetic disease that has been diagnosed. So, I mean, what's going on? Is it the same that the cells, they become different? So as we collected aneurysms, we could also collect the cells of people having these aneurysms and test them. And so what we did is that we did traction force microscopy. So traction force microscopy, so this has been done a lot here in Barcelona by the group of Xavier Ferrat, for instance, and we used like the principles that they developed. So we culture the cells on gels, and these gels, they have beds that we can track the motion of these beds. And you see here, for instance, that if you have a cell, and if you have a cell that is cultured on this gel, and you see that we apply a chemical that will just kill the attachment of the cell to the gel. So you see the cell will just relax, but see also that here the beds then move, okay? And so we can track the motion of these beds like this, and with that, we can measure the forces that are applied by the cell on the gel. So these forces, they will tell us, is the cell applying more or less force than, well, in an aneurysm or in a normal patient, for instance. And so the question was, what would you guess, actually? Would you guess that if I take the cells from an aneurysm patient, the cells would be stronger or they would be weaker? They would apply bigger forces or smaller forces? Smaller for me. That's what we thought as well, okay? Because we were talking with biologists and they said, okay, the cells, they cannot maintain the wall, okay? If the cells would be strong enough, they would maintain the wall. So we tested many cells actually, and actually the first thing is that we discovered, and now it has been confirmed with by other analysis which are more related to the gene expressions, that the smooth muscle cells that we have in the wall of an artery, it's like the human population. Okay, it's like everyone in this room is different, so all these cells are different, so none is going to apply the same force. And we have like for a given aneurysm or a given non-aneurysm healthy aorta, we have quite a wide range of variations of the forces, but we found a trend of finding cells that would be stronger in the aneurysm. And these cells actually are like among us, would be like maybe like the guys who do the gym every day, for instance. I don't know, but in the aneurysm, actually, these cells, they develop for some reason that actually they represent myofibroblasts, for instance, which is a type of cell that will be an evolution of the smooth muscle cells that relates to repairing the wall. And another type also that has been observed, and for endothelial cells, for instance, has been observed like senescent, so cells that become senescent also, because it's also usually in aged people. So senescent cells apply stronger forces, but we were not able still for smooth muscle cells. Nobody has ever found that senescent cells would be stronger. So we're still working on that. But so we have stronger forces for the aneurysm cells. So we found that it's more like myofibroblasts that are expressed, and that these cells that need to repair the wall. We also checked if the cells, they change, and so we did also AFM indentation to measure if the cells, they are stiffer or softer, but we didn't find a big change between the cells from healthy aorta and the aneurysm. And the thing is that we probably have also a lot of variations in our cells, because these cells, they are cultured on a gel in 2D, and so in reality, their conditions are very different. So we are trying now with different postdocs to mimic more in 3D hydrogels with a special microstructure, but that we try to design the condition of these cells that then we could still use in these conditions the same process of releasing their forces and measuring the deformation. So we have developed a device with optical coherence tomography to track the deformation in 3D in a gel and then measure these forces. But it's still a hot topic for us at the moment. And well, but still, I mean, every time, so we were like putting the finger in mechanobiology, and then now we have the arm, and I guess like it's getting more and more, because the question is still, well, but what's going on? I mean, now we have all these cells, and so does the cell population change during life? And that some people, they develop these some of these cell phenotypes that are stronger, and do they develop them because of this stiffness that we found in the aneurysm, that then they are less stimulated because the wall is less pulsating, and so then this could also contribute to the aneurysm. So we started to get interested in this cell phenotype. So the phenotype of the cell, so it means like all the traits, so like all the features that can be related to defining the function. And in a cell, you have this nucleus, and the nucleus, it will express genes, okay? And depending on the cell type, you will have different genes expressed. And some cells, like the contractile smooth muscle cells, they will express the genes for these proteins of their contractile apparatus, that is like the myosin, for instance. But you have also like adipocytes, which are the cells of the fat. You have osteogenic cells, like are the cells of the bone, and you have macrophages, which are the cells of the inflammation, fibroblasts, which are the cells that produce collagen, mesenchymal cells, which are stem cells. So some people, they found, this was the first paper showing that, they are all inside the wall of the aorta. So the smooth muscle cells that we found were like a unique type. Actually, it's, I mean, we have all these cell types. And so we thought, okay, so does this population change if we change the mechanical condition? And this would explain also why then the aneurysm evolves. And so then we started a new experiment where we mimicked an aorta where it would become so stiff that it would not pulsate, and then another one where it would have a pulsation of 10%, so like a stretch at 1 hertz. And then we cultured the cells on these two with a system that is actually it's a rubber membrane, and the system is a flexen, and we culture the cells on them. And then after a given time, we extracted the cells, and then we did some qPCR analysis to look at the genes that were expressed. So the qPCR is, you know what they did with for the COVID test. Okay, so it's a proteomic analysis, but so we can also characterize the genes that are expressed. And what we found that is something very interesting that now we are also trying to see how this could be integrated, is that a number of genes that relate especially to a production of matrix in the aorta, they evolved. So their expression increased from 0 day to 3 days in this experiment, but it never stopped increasing, but very slowly. But all the same genes in the mechanostimulated experiment, they were expressed from the beginning at a given level, and this was maintained all along. Okay. So what we understand by that is that actually when there is the mechanostimulation, then there is a regulation, a mechanoregulation of the gene expression. So the cells that are maintained, like the phenotype is maintained. And then when we remove the stimulation, then the phenotype may change. So this is a lot of factors that may combine together to contribute to maintaining or not maintaining the phenotype. But we can see here that the mechanics contributes a lot. So this goes back to this beginning when I said, okay, so we first started to observe that the region where we had high stiffness and where we had no wall shear stress vector, this was where the trouble started. Okay, and then this closes the loop because we see that actually no mechanical stimulation, lack of mechanical stimulation, then this will induce that there is a lack of mechanoregulation down to the gene level. And this can be explained by this mechanotransduction principle. But so then with all that, what can we do now? So what could we do in the future? So I go back again to the very beginning of my presentation. So I told you 2010, the surgeon came. So we need to do a computer model and we have a startup and then we can stimulate the medical device. Okay, I think this worked, but as I said, this was using like technologies and scientific results that were not really up to date, but it works and it's helpful now for the surgeons. I think that in the future, we can have digital twins that are multi-scale and that can take into account observations that are at the organ level, translate them at the cell and subcellular level in order to induce the response in terms of regulation equations and predict the remodeling and then predict the next stage of the aneurysm. So this will become possible, but this remains complex. And if this is possible, this is also possible to have a virtual twin of someone. And we can, for this someone, say, okay, if I try this drug, what's going to be the effect? Or if I try that drug, what could be the effect? Which would be very useful for the aneurysms because now it's still a disease where there is no drug. There were some attempts, but the attempts are, if you have an idea of a drug, now the only thing you can do is you go and you make a clinical trial. And this clinical trial means you find people who have an aneurysm on who you are going to try a drug. Okay. So the idea with these virtual twins is that you would create a virtual cohort and then you could try these drugs. And so this would probably speed up the discovery of these drugs for the future possibility of treating this aneurysm by arresting the aneurysm growth and not like having a palliative stem graft deployment in the aneurysm. That's one of the main prospects. So we're working on that. So our multi-scale now, we're developing it by having computer model solving the mechanics at different scales, and we're working now at that scale where we have the cell also modeled in terms of mechanics. And well, I just show you, for instance, one of these models that was developed by a postdoc. So this is a model of a cell where we solve the equation. So we included different components of the cell, the nucleus, the cytoskeleton, the cytoplasm, the fibers in the cytoskeleton, and the membrane, and so on. And the idea is to be able to investigate how the nucleus of the cell is loaded in these situations, because as I said, in this mechanotransduction axis, everything goes down to the nucleus. And so we need to see in which situation the nucleus will be loaded or less loaded. And you see here, for instance, in this simulation that I showed, that when you have the fibers that are contracted, then there is the nucleus that is squeezed. And so then this is a situation of load of the nucleus that we try to understand, because the last thing I should say is that there's another genetic disease that is called progeria. So this is the hardest disease possible. This is a disease where the people, they die at age 10 because they have a problem to the protein that makes the envelope of the nucleus. And the envelope of the nucleus is made of a protein, the name is lamellae. And if there is a defect, then the nucleus becomes brittle, and so it makes some bulges, and then the DNA can go out, and then the cells, after they, to auto-protect them, they just go on apoptosis. So do you know what first disease these people develop? It's an aortic aneurysm, and they die of aortic aneurysm. So because of the lack of lamellae, they also develop an aneurysm. So that's why we try to relate all these scales together, and most of the science we use is mechanics, and so that's why I was coming here to talk to you. I thank everyone contributing to this work. I may probably miss someone, but overall, I thank you for your attention.
Thank you very much, Stéphane, for this nice presentation. We have time for questions. Someone wants to start?
So thank you a lot for the interesting talk. So in this connection between what happens at the cellular level and the disease, let's say, is there like an intermediate experimental system that is not an animal or a patient? You know, in a organoid like vein, where you can have like more controlled experiments and measurements that will, because it's, I mean, you very interesting work, but you notice that it's very challenging to really, at the end, you did this simulation of how initial simulation of how the position of different components of ECM would explain your phenotype and so on. But it looks like very challenging to be honest, right?
Yeah. So 1 year ago, I would have thought that my answer would have been this would create even more problems, because I mean it's very challenging to develop these organoids. There are different challenges related to the material you choose, and a lot of chemistry and so on. But then we came to the same conclusion as what you say, that this would be very useful, and so I think this does not exist. So we submitted different grant proposals to build these collagen hydrogels with guys, specialists of collagen hydrogels, that we could still at least mimic in vitro the conditions of smooth muscle cells that then we could study in better conditions than, like, because you say it's a too big jump. You have a patient, you have a tissue that you can observe, and then you have the cell culture on 2D on the membrane. Okay, so it's I mean, it's too big jump, and then you want to develop numerical models from that. I agree with you, there's something missing, so we try to work on that. But as far as I know, nothing like you may find for other organs like kidney or liver, for instance, where people have developed these organ-on chips. Like vessel-on chips exist, but it's more for endothelial cells and not for what's inside the wall of the smooth muscle cells at the moment. It's not mature for smooth muscle cells. So we try to work on that.
Thank you for the talk, very nice. And I was wondering, when you describe this interaction between the large scale and the micro scale, and so I more or less realize what you do from the large scale to the micro scale. But how do you account? I mean, it's already ongoing, this type of models that take into consideration the phenotypes and the mechanical property, or something that is still on the...
Yeah, so I would say that for me, this is what I found, like mechanics has already solved. Like if I know what are going to be the ingredients at the micro scale, then I have tools in mechanics that can upscale and homogenize the behavior. But of course, you need to know all the ingredients, not only the fiber, their behavior, but also their interaction, which is, I think, still complex. But the other way around, for me, is completely now. I mean, a challenge is how. I mean, the stresses that you have, and also this even relates to the definition of stress in continuum mechanics, because stress is like that you model an artery at the continuum medium, but then after you translate that to the cell level, so you have to change the scale, and so then what kind of mechanical loading you have at the cells, and then down again inside the cells. And does it matter? What parameter matters? And it becomes also so variable. So then you have to take into account statistical variability that is also another aspect that I didn't talk about. But yeah, I find that maybe it's important to solve first in this direction, because the other direction, I think there has been a lot of literature about homogenizing things up.
But homogenizing from the mechanical viewpoint, I understand it, but if you have to incorporate information from the genotype. Okay.
Yeah, but this information about the phenotype of the cell is, I mean, they are important for two aspects. One is first, if you know it, it's important because it tells you the function. And let's say that, for instance, in an artery, I know that there's going to be more and more cells that become senescent. So this is a phenotype. And then if I know it, then I know, okay, I have to do something. Okay, so I don't want to know what is going to be the consequence at tissue level, but maybe first for the biologist and for the clinician, they know that they need to do something. Okay, that's already something important for them. If we can predict the function of the cells, that may be why I was also more focusing on that direction of multi-scale translation.
Questions?
Yeah, I mean, when you have an aneurysm in a rat, something that you can, heal to test the material or something, is it possible experimentally to estimate of the tension that the aortic wall is bearing, which fraction comes from the contraction of the muscle cells, which part comes from the deformation of the ECM?
There was so on rats, or even was done in vitro, like some tests were done even from I mean tissue coming from human, but from rats, for instance, you can do, and this was done a long time ago. You can use a chemical like elastase to remove the elastin, then collagenase to remove the collagen, and then you see what's the response if you do one treatment or the other. But this, I mean, there's no one component independent of the other. So everything works in synergy. So that there are cross links, and there are also small components like I mean, glycosaminoglycans are small molecules that participate to the link between all this. So I don't know. I mean, maybe you have a different opinion, but I think that nothing can be. Okay.
You do like a filter, you don't destroy.
Yeah, but the thing is more about the cells. So the thing is that when we do a test on the cells, so we usually have the cells already dead. If they would survive fresh tissue, maybe they would survive 3 hours. Okay, so I think most of the literature on these tests and the data we're using, the cells are dead. Okay, and so well, the contractile apparatus of the cells is probably more contracted than it is in vivo, and also in vivo, like the cells, they I mean, every time, this is something that has not been investigated enough, I believe, is that the cells, the smooth muscle cells, with the pulsatile behavior of the artery, so they are stretched and unstretched all the time, and then this will induce, I mean, flux of calcium through their membrane all the time. In, out, in, out, in, out. And then this will change their tension. And so this is something that we don't even know if, like, the cells, they change their tension all the time, because we cannot measure that at the 1 Earth in vivo. But that would be something important to know, I think, more than the cells after removing the artery. I think that people have done like contracting the cells, not contracting, but what is the actual behavior in vivo, I think is still something important to characterize.
It's just to probably this helps a bit, I'm not really sure, but one thing you can know is more or less the density of this type of cells inside the tissues, depending on how far from the heart, and also the importance of the collagen here with the smooth muscle cells in the contractile effect is very, in the stretchiness, because it's very important. So if you measure the density of this type of components in the vessel, you can more or less assume some type of different behavior according to this. But yeah, you have to do everything with histologies and all things like that.
So what do you think of modeling, like mechanically at that scale, like the cell scale now? What do you think?
It's a very important thing. Actually, the effect of the deformation in the cell differentiation is critical, because the deformation of the cell directly goes to the nuclear envelope, the one you were talking before. So this is what provokes this exchange of the gene expressions and all that stuff. So these are really, really good topics for analyzing this area.
Yeah.
Yeah.
But that was a good question from Stéphane. Yeah, it's true that now already, if we know then what would be the consequence on the cell function, I think we already have something to say, because a drug, for instance, will target the cell function. Okay? And then people are looking for drugs that will induce a specific response in terms of cell function. The problem is that this response can change the mechanics and then, or change, affect another system like of the body, and then it has like so many side effects that in the end it does not work. And so that's the kind of thing that we would like to predict with these models, because otherwise this will be trial and error with clinical studies.
And for this ambitious future digital twin, what type of inputs would you use? So I understand the imaging, where you capture this deformation and the stiffness distribution and so on, but then we also need information like the proteomics of tissue, or what is it?
Yeah, so the proteomics and the transcriptomics would tell us, I mean, we would constitute a database to relate like the induced function depending on the different environment that can exist. So this would be data to train the digital twin to predict the response. But then the condition of the cells, the mechanical conditions, we would like to compute them. Okay, so and then if we know like the mechanical conditions that we can compute, and then other conditions that we can maybe assess, and then we have a database of transcriptomics that tells us what's the response of the cells depending on these conditions. Then after, for a given patient, we we can do some predictions. But at the moment, I think transcriptomics is like kind of it's the blossom of transcriptomics, like can be a single cell and so on. And so there would be so many discoveries because at the moment I show you transcriptomics, but it pulls like a response of thousands of cells. Okay, and so as I said, like the cells are probably the same as we are. Okay, I mean so different, and some of them they in the world when the elastin starts to be damaged, some of them they have no more stimulation, others is still like the elastin is almost intact. So some of them they live in a world that is the same as when we are 20 years old, and others they are already like close to the end of the life. And at the moment we don't know how this affects and how to detect. I mean. The fraction of these populations in the world. That for me, that's going to be very important when we can do that. Then, if we take an image of the aorta of someone and we see like its distribution of structure, okay, we see that the cell functions in this aorta, we are still a lot of cells that we can support that they can still continue to maintain the wall and overlook. And here we need to support the cells. We need to give that drug that will maybe restore the contractile apparatus of the cell. But this kind of thing that we would like to do with the digital twin.
Is there any other question? From the people connected, I don't know if there is a way I completely forgot. Somebody wants to ask one question. Okay. Otherwise, well, Stéphane will be here for the rest of the day and tomorrow also, the afternoon. So, well.
Thank you. And I'm happy to talk about the finite elemental resolution of this because, yeah, I know a lot of you are really, I mean, very strong in like all the computer resolution of these models that, well, it created a lot of issues for us, and we still have maybe some issues also with compressibility and so on. And well, it's better for me to talk about things I'm kind of let's say comfortable with in the talk. I think that's even what I always tell to my students: don't talk too much about things you're not comfortable with, because then this. But this is also an invitation to more discussion, I would say. And also, I would like to say that I plan to open positions of postdocs. So if you have some PhD students after a PhD here would like to go to France for a postdoc experience, there will be position opening, and especially one position would be in developing this model at the cellular level that I was mentioning. So if anybody here, or if you know anybody that would be interested by such a topic, just pass the word, and just a person can just send me an email. The position is not advertised yet, but it's already open.
And maybe briefly, we are organizing a summer school in the context of this project for the next year between Saint Etienne and Sydney, and details in the next months. Yeah. Okay, thank you very much again, Stéphane.